All right, so welcome back to the deep dive. Now, if you've been with us for our GCSE biology series, you'll remember that in our last deep dive, we were looking at homeostasis and how your body keeps everything in balance. Well, for episode 6 out of 7, we're kind of building on that a bit. We're going even deeper into how living things pass on their characteristics and how life itself has changed over a really long time.
Yeah, that's right. So basically, you're joining us as we look at inheritance, variation, and evolution. And it's kind of like a need-to-know guide for your GCSEs.
Perfect.
We know it's a math topic. We're talking about things from the tiniest molecules all the way up to the history of life on Earth. So what we want to do is make sure you understand the really key ideas without getting bogged down in too much detail.
Okay, well, let's start right at the beginning then with reproduction. I mean, it is how life goes on, right? And there's basically two ways organisms do this. Can you explain those for us?
Yeah, sure. So the first one is sexual reproduction. This normally involves two parents combining sex cells or gametes.
Okay.
If we're talking about animals, that's the sperm and the egg cells. And for plants, it's pollen and egg cells. Now, here's the interesting bit. These gametes don't actually have the full set of chromosomes. They only have half.
Right.
So a normal human cell has 46 chromosomes in 23 pairs.
Okay.
But a sperm or egg cell only has 23 single chromosomes.
So when those two combine, that's what we call fertilization.
Exactly. Fertilization is when the nuclei of the male and female gametes fuse together.
Okay.
This is important because not only does it restore the full number of chromosomes back to that 46, but it also mixes the genetic information from both parents.
Right.
And this mixing is what causes the variation we see in offspring.
That makes sense. So that's sexual reproduction. What about the other method, then, the one that doesn't need two parents?
So that's called asexual reproduction.
Right.
This only needs one parent. And there's no fusion of gametes at all. Instead, it uses mitosis to create new cells.
Okay.
And you might remember from your studies, mitosis is when a parent cell divides to produce two identical daughter cells.
Yeah.
So the offspring are basically clones of the parent, like bacteria splitting, or strawberry plants sending out runners, or daffodils growing from bulbs. And, even some animals can reproduce asexually too.
So if sexual reproduction is all about mixing and creating differences, how do those gametes, the sperm and egg, end up with only half the chromosomes and that potential for so much variety?
So this is where we have this process called meiosis.
Okay.
Now, meiosis is a bit different from mitosis.
Right.
In meiosis, the cell first duplicates all of its chromosomes. So for a bit, it's got double the genetic information. Then it goes through 2 rounds of division, not just one. And we end up with 4 daughter cells. And these are our gametes.
Okay.
Each one with half the original number of chromosomes, so 23 in humans.
Okay, 4 cells, each with half the genetic material. But where does the actual difference come from? You mentioned something about shuffling earlier.
Yeah, so during those 2 divisions, the chromosomes are shuffled around and they're randomly sorted into those 4 daughter cells.
Right.
Think of it like dealing cards. Each gamete gets a different hand.
Okay.
So each gamete ends up with a unique mix of the parent's genetic material. And this shuffling is what ensures all the gametes are different from each other. And this leads to even more variation when they finally fuse during fertilization.
So 2 very different ways of making new life. What are the advantages and disadvantages of each? Why would an organism use one over the other, or sometimes even both?
That's a really good question. So the big advantage of sexual reproduction is variation in offspring.
Okay.
Now, this is really important because if the environment changes, like say there's a new disease or the climate changes, then there's more chance that some individuals in that population will have traits that will help them survive and reproduce.
Right.
So some might not make it, but because there's so much variation, it means the whole species is less likely to die out.
Right.
And sexual reproduction also lets us do selective breeding, which is where we intentionally breed organisms for characteristics we like.
Right, so like farmers breeding sheep for thicker wool or scientists developing wheat that's more resistant to drought.
Exactly. Now, asexual reproduction has some benefits too. It only needs one parent, which is helpful if it's hard to find a mate or the population is small.
Yeah.
It's generally quicker and uses less energy because there's no need for complicated mating rituals or all those special reproductive structures.
Right.
And in a stable, unchanging environment that's good for the parent, asexual reproduction is great for producing lots of offspring really quickly that are all perfectly suited to those conditions.
So it's like picking the right tool for the job depending on what's going on around them.
Exactly. And it's fascinating that some organisms have evolved to use both methods.
Oh, really?
Like the malarial parasite, it reproduces sexually in the mosquito, which is its main host, but asexually when it's in humans.
Okay.
And some fungi usually release asexual spores to spread quickly, but they switch to sexual reproduction when the environment changes to make sure they survive.
Clever.
And plants like strawberries use asexual reproduction to spread quickly with their runners, but they can also reproduce sexually using flowers and seeds.
Right.
This double strategy is especially useful for plants because it means they can reproduce even if their flowers get damaged.
Makes sense. Okay, so I've talked about how organisms reproduce and the role of meiosis in creating variation through sexual reproduction. Now let's get into the actual instructions for life, DNA, and the genome. What are they exactly?
So think of DNA as the chemical that makes up the genetic material in the nucleus of pretty much every cell.
Okay.
It's a really amazing molecule. It's a double-stranded polymer that's all twisted up in a double helix shape, like a spiral staircase.
Uh-huh.
And this DNA is organized into structures we call chromosomes. Now, a gene is a section of DNA on a chromosome. It's like a code for building a specific protein.
Okay.
So a protein is made up of lots of amino acids joined together in a specific order.
So genes are like the recipes for proteins, and proteins do all sorts of jobs in the body.
That's a great way to put it. And the genome is the complete set of instructions.
Right.
It's all the genes that make up all the proteins an organism can produce. The Human Genome Project was this huge undertaking that mapped out the entire human genome. It's been incredibly important.
Oh, wow.
We now understand so much more about how our genes are linked to diseases, inherited disorders, and even how humans have migrated across the world. It's really foundational stuff for all sorts of medical advancements.
Now, for those of us aiming for the top grades, can we go a bit deeper into the structure of DNA? Like, what's it actually made of?
Yeah, sure. So DNA, which stands for deoxyribonucleic acid, is a polymer, which means it's made up of repeating units called nucleotides.
Okay.
And each nucleotide has three main parts: a sugar molecule, a phosphate group, and one of four bases.
Okay.
So these bases are adenine, cytosine, guanine, and thymine, or ACG&T for short. The sugar and phosphate groups make up the backbone of the DNA strand.
Right.
And the bases stick out and they pair up with bases on the other strand.
Okay.
But the pairing is specific. Adenine always pairs with thymine, and cytosine always pairs with guanine.
Okay.
This is called complementary base pairing. And the order of these bases along the DNA strand is what makes the genetic code.
So it's like a language.
Exactly. And each sequence of three bases codes for a particular amino acid.
Right.
Now remember, not all DNA actually codes for proteins. There are sections called non-coding DNA, and these are really important for controlling when and how genes are expressed.
So they're like the on and off switches.
Exactly, like punctuation in those recipes telling the cell when to start and stop reading a gene.
Right. Okay, so the DNA has the code in the sequence of these bases, but how does that code actually get turned into a working protein?
So that's called protein synthesis.
Right.
And it's basically the process of using the instructions in the DNA to build proteins. Now, the first step is that the DNA is too big to leave the nucleus. So its instructions have to be copied onto a smaller molecule called messenger RNA, or mRNA.
Right.
This mRNA is made using complementary base pairing with one of the DNA strands. Once the mRNA template is made, it leaves the nucleus and goes into the cytoplasm where it meets up with ribosomes.
Okay.
Ribosomes are the protein-making factories of the cell. And at the ribosome, the sequence of bases on the mRNA is read in groups of three.
Right.
Each group, called a codon, specifies a certain amino acid. Then another type of RNA, called transfer RNA or tRNA, brings the correct amino acids to the ribosome based on what the mRNA says.
Okay.
These amino acids are then joined together to form a chain. And finally, this chain of amino acids folds up into a unique 3D shape.
Uh-huh.
And it's this shape that determines what the protein does, whether it's an enzyme or a hormone or a structural protein.
So even a small change in the DNA code, like swapping one base for another, could mess up the whole protein building process.
Absolutely. And that's where mutations come in.
Right.
A mutation is a random change to the DNA sequence.
Okay.
These changes can happen in a few ways. A base can be added, a base can be deleted, or a base can be swapped for a different one. And because the genetic code is read in groups of three, adding or deleting a base can really throw things off.
Right.
It can change all the amino acids after that point. Swapping a base might only change one amino acid, or sometimes it doesn't even change anything, because the genetic code has some redundancy built in.
Okay.
But if the amino acid sequence does change, it can affect how the protein folds and therefore how it works.
Makes sense.
Most mutations have no effect, or a very small effect. But sometimes a mutation can have a bigger impact, like making an enzyme not work, or changing the shape of a structural protein. And like we talked about before, Mutations in non-coding DNA can also change gene expression.
Right.
So all the variation we see between different organisms is due to these differences in both coding DNA, which makes proteins, and non-coding DNA, which controls when and where those proteins are made.
So we're starting to see how inheritance works at the molecular level. Now, what about how these genes are passed down from one generation to the next? What do we need to know about genetic inheritance?
So there's some key terms we need to get familiar with. It's a bit like learning a new language.
Okay.
We've talked about gametes already, those sex cells with half the usual chromosomes. And then there are the chromosomes themselves, which are those long strands of DNA. And a gene is a section of DNA that codes for a protein.
Right.
Now, lots of genes have different versions. We call these different versions alleles.
Okay.
So think about eye color. There might be an allele for blue eyes, another allele for brown eyes.
Right.
Some alleles are dominant, which means that if you have even just one copy of that allele, its trait will be expressed.
Okay.
We usually show dominant alleles with a capital letter. Recessive alleles are only expressed if you have two copies.
Okay.
And we use a lowercase letter for those.
Right.
If someone has two identical alleles for a gene, either two dominant or two recessive, we say they're homozygous for that gene.
Right.
If they have one dominant and one recessive allele, they are heterozygous.
Okay.
So the combination of alleles an individual has is called their genotype.
Right.
And the characteristics we actually see, like their eye color or blue type, is their phenotype.
So genotype is the genetic makeup, and phenotype is what we actually see.
Got it.
So how do we actually track how these alleles and traits are passed down through families? I've seen diagrams that look like family trees.
Yes, exactly. Family trees or pedigree charts are diagrams that show how traits are inherited over several generations.
Right.
They use symbols for individuals and lines to connect them, showing relationships and whether or not individuals have a particular characteristic.
Okay.
And to predict what genotypes and phenotypes the offspring might have from a particular cross, like between two parents with known genotypes, we use something called a Punnett square.
Right.
It's a simple grid that shows all the possible allele combinations the offspring could get.
Yeah, I remember those squares. We should also talk about inherited disorders. How do they fit into all this?
So inherited disorders are diseases caused by inheriting faulty alleles.
Okay.
A good example is polydactyly, where someone is born with extra fingers or toes.
Right.
Interestingly, this is caused by a dominant allele. So if you inherit even one copy of the faulty allele, you'll have the condition.
Okay.
Another example is cystic fibrosis, which affects the cell membranes and causes thick mucus to build up.
Right.
Now, cystic fibrosis is caused by a recessive allele, so you'll only have the disease if you inherit two copies of the faulty allele, one from each parent.
Right.
If you only have one copy, you're a carrier. You don't have a condition, but you can pass on the faulty allele.
And all this knowledge about inherited disorders has led to things like embryonic screening, right? What's involved there?
So embryonic screening is a way of checking embryos for faulty genes that cause inherited disorders.
Okay.
It's usually done with embryos created through IVF.
Right.
They take a few cells from the embryo when it's really early in development, and they analyze the DNA to see if there are any alleles for particular genetic disorders. They can also do similar testing on a fetus while it's still in the womb.
Okay, so that explains what it is. Now, before we move on to evolution, let's talk about how the sex of a baby is determined in humans. It's not just down to one gene, right?
No, it's not. The sex of a human is determined by a special pair of chromosomes called the sex chromosomes.
Okay.
These are the 23rd pair. We have 22 other pairs called autosomes, which control all our other characteristics. But the sex chromosomes are either X or Y.
Right.
Females usually have two X chromosomes, so XX, and males usually have one X and one Y, so XY.
Okay.
When gametes are made, females can only pass on an X chromosome in their eggs, but males can pass on either an X or a Y in their sperm.
So it depends on the sperm.
Exactly. If a sperm with an X chromosome fertilizes an egg, the baby will be XX in female. If a sperm with a Y chromosome fertilizes the egg, the baby will be XY in male.
Okay.
So it's roughly a 50-50 chance of having a boy or a girl.
Right, that makes sense. Okay, let's talk about variation and evolution. We've already touched on how variation comes from sexual reproduction and mutations, but how does the environment affect things?
So it's not just genes that determine an organism's phenotype, which is those characteristics we actually see.
Okay.
The environment an organism lives in also plays a big role in how those genes are expressed. Think about a scar on an animal. That's a physical characteristic, but it's not genetic.
Right.
Or the color of a plant's leaves. The genes tell the plant how to make chlorophyll, but the amount of sunlight affects how green the leaves are. Leaves in the shade are often paler.
Yeah, that's true.
And lots of traits are influenced by both genes and environment, like a person's height. It's partly genetic, but also depends on their nutrition.
Right.
Same with weight. Your genes affect your metabolism, but your diet and lifestyle have a big impact too.
That makes sense. We mentioned earlier that mutations can introduce new variations into a population. Are all mutations important for evolution?
No, not all of them. Most mutations have no noticeable effect at all.
Right.
Some might cause a tiny change, but it's rare for a single mutation to create a whole new advantageous characteristic.
Okay.
However, if a mutation does give an organism a survival advantage in its environment, things start to get interesting.
Okay.
So if a mutation helps an animal camouflage better, or find food more easily, or attract a mate, then natural selection comes into play.
Ah, yes, Darwin's big idea. Can you remind us what the theory of evolution by natural selection is all about?
Sure. So the theory basically says that all the diversity of life we see today has evolved over millions of years from simpler life forms that existed billions of years ago.
Okay.
And the main driver of this change is natural selection.
Right.
The idea is that in any population of a species, you have natural variation, and a lot of this comes from random mutations.
Uh-huh.
Some of these variations give individuals an advantage,
Okay.
like maybe they're faster or have a better sense of smell or can digest a certain food better.
Right.
These individuals are more likely to survive, reproduce, and pass on their advantageous genes.
Okay.
Over many generations, the frequency of these good alleles increases in the population. If this goes on for long enough, and if populations become isolated and face different pressures, it can lead to speciation,
Right.
which is where you get new species that are so different they can't interbreed anymore.
So it's about survival of the fittest, but fittest really means best suited to the environment.
Exactly. And it's important to remember that natural selection doesn't act directly on the genes.
Okay.
It acts on the phenotypes, the characteristics we see. But because those phenotypes are mostly determined by the genes, it's the frequency of certain alleles that changes over time.
Now, humans have been intentionally influencing the traits of plants and animals for thousands of years through selective breeding. How is that connected to natural selection?
Selective breeding is like artificial selection. Instead of the environment deciding which individuals are most likely to survive and reproduce, humans choose organisms with traits they like and breed them together. We've been doing this with animals for ages to get breeds that produce more milk or have a better temperament or are good for certain tasks.
Huh.
We've also selectively bred crops to make them more disease resistant or to give them higher yields or bigger fruits.
Right.
And the way we do this is by carefully choosing parent organisms with the traits we want breeding them together, and then from their offspring choosing the best ones to breed again.
Okay.
We keep doing this over many generations, and each time we get closer to the characteristics we're looking for.
So, are there any problems with selective breeding over a long time?
Yeah, one issue is that it can increase the risk of inbreeding.
Okay.
When we keep breeding individuals with similar traits, we're also breeding individuals that are close to related.
Right.
This can reduce the genetic diversity or the gene pool.
Right.
And a smaller gene pool means fewer different alleles, which can make the population more vulnerable to changes in the environment or new diseases.
So it makes them less resilient.
Exactly. And inbreeding also increases the chance of harmful recessive alleles being passed on, which can lead to more genetic disorders.
Right. So by focusing on a few traits, we're accidentally making them weaker in other ways. Now let's talk about genetic engineering. How is that different from selective breeding?
So genetic engineering is a much more direct way of changing an organism's characteristics. Instead of gradually changing the gene pool through breeding, genetic engineering involves actually manipulating the organism's genome by inserting a specific gene or genes from another organism to give it a desired trait.
So it's like cutting and pasting DNA.
Kind of. So, for example, scientists have genetically engineered plants to be resistant to certain pests or diseases or to produce more vitamins.
Okay.
And bacteria have been genetically modified to produce useful things like human insulin for treating diabetes.
Wow, that's amazing. Can you explain how scientists actually insert genes into another organism?
Yeah, sure. So first they have to identify and isolate the specific gene they want. They use special enzymes called restriction enzymes to cut the gene out of the DNA of the donor organism.
Right.
These restriction enzymes cut the DNA in a staggered way, leaving sticky ends which can easily join to other complementary sequences.
Okay.
Then they use a carrier molecule, often a circular piece of DNA called a plasmid or a virus, and they cut this with the same restriction enzyme to create matching sticky ends.
Uh-huh.
The isolated gene and the cut vector are mixed together, and another enzyme called DNA ligase binds them together.
So that inserts the gene into the vector.
Right. Now, this combined vector is introduced into the host organism's cells.
Okay.
So for GM crops, this might be into plant cells. If it works, the host organism will incorporate the new gene into its own genome and hopefully express that desired characteristic.
So it could mean crops that are resistant to insects or herbicides, which would increase yield.
Exactly. And in medicine, they're using similar techniques in gene therapy to transfer working genes into patients' cells to treat genetic disorders.
Right, so it's like borrowing an instruction from one organism and giving it to another. Now, I know there are lots of different opinions about genetic engineering. What are some of the potential benefits and risks?
Yeah, it's a big topic with lots of different viewpoints. The potential benefits are huge. Things like being able to produce medicines and vaccines using genetically modified organisms,
Right.
or developing crops that are more nutritious or resistant to pests and diseases, which could help with world hunger. And then there's gene therapy, which could potentially cure inherited diseases.
Wow.
But there are also risks and ethical concerns. Some people are worried about the impact of GM crops on the environment,
Okay.
things like how they might affect insects like bees or whether those engineered genes could spread to wild plants. There are also questions about the long-term effects of eating GM foods on human health,
Right.
although a lot of research suggests that the GM foods we have now are safe. Ethically, some people are uncomfortable with the idea of altering an organism's genes and worry about unforeseen consequences.
Okay.
And then there are the debates around designer babies and whether everyone would have access to these technologies. In agriculture, there's also the concern that using herbicide-resistant crops could lead to herbicide-resistant weeds, which would mean having to use even stronger chemicals.
So it's a powerful technology, but we need to be careful.
Exactly. It's a field with massive potential to help people, but we need to think carefully about the ethical and environmental implications.
Right. Now let's touch on cloning. We know asexual reproduction makes clones naturally, but how do humans intentionally create clones?
So cloning is basically creating genetically identical copies of an organism.
Okay.
With plants, there are a couple of fairly simple methods. One is tissue culture, where you take a small sample of plant cells and grow them in a special nutrient-rich medium with hormones.
Okay.
These cells can then develop into whole new plants that are genetically identical to the parent plant. Another common method is taking cuttings.
Right.
So you might take a stem cutting from a plant with good traits, dip it in rooting powder, and plant it. And then it can grow roots and become a new plant that's a clone of the parent.
What about cloning animals? That sounds more complicated.
It is. The simplest way is embryo splitting, which is basically artificial twinning.
Right.
So you fertilize eggs with sperm from parents with good traits. The embryos develop for a little while, and then you split them into two or more identical groups of cells.
Okay.
Each group is then implanted into a surrogate mother, and if the pregnancies are successful, the offspring will be genetically identical because they came from the same fertilized egg.
Right.
A more advanced technique is somatic cell nuclear transfer, or SCNT. This is what they use to clone Dolly the sheep.
Oh, wow.
They take the nucleus from an adult body cell of the animal they want to clone and put it into an egg cell that's had its own nucleus removed.
Right.
Then they stimulate the egg cell to divide, and if it develops into an embryo, they implant it into a surrogate mother.
And that creates a clone of the donor animal.
Exactly. So what are the potential benefits and risks of animal cloning?
Right. What are those?
Well, one benefit is that you can quickly produce lots of offspring with specific desirable traits, which is useful in agriculture and research.
Okay.
Studying cloned embryos can also help us understand early development, and some scientists hope that cloning could help save endangered species or even bring back extinct ones if we have their DNA.
Wow.
But there are also risks. Cloned animals often have more genetic problems and health issues, and they may not live as long.
Right.
Cloning is also quite difficult and has a low success rate. And if we use cloning a lot, it could reduce genetic diversity, which would make populations more vulnerable to diseases and environmental change.
Okay.
And of course, cloning humans is a whole other ethical can of worms.
Definitely lots to think about there. Okay, let's look back at how our understanding of genetics and evolution has changed over time. It wasn't always this clear, was it?
No, definitely not. What we know today is the result of the work of loads of scientists over centuries building on each other's discoveries. A really important figure is Charles Darwin. He published his theory of evolution by natural selection in 1859 in his book On the Origin of Species.
Right.
His theory was based on his observations of variation, the struggle for survival and inheritance. He said that organisms best suited to their environment are more likely to survive, reproduce, and pass on their traits.
Yeah.
But his ideas were controversial at the time.
Why?
Well, they went against religious beliefs about creation. And back then, no one knew how traits were inherited or where variation came from.
So there is no scientific explanation.
Not at that time. There was another theory of evolution going around called Lamarckism.
Okay.
Jean-Baptiste Lamarck, another scientist, thought that changes an organism acquired during its lifetime could be passed on.
Oh, right.
Like the idea that giraffes got their long necks from stretching to reach leaves.
Yeah, I've heard that one.
But that theory was disproven when we understood more about genetics.
Right.
Darwin's theory became more accepted as we found more fossils showing the progression of life. and as genetics gave us a way to explain inheritance.
Okay. You mentioned speciation earlier, the process of new species developing. How did we come to understand that?
So speciation basically comes out of Darwin's theory of natural selection.
Okay.
It happens when populations of a species become separated, often geographically.
Right.
They face different environmental pressures and different appeals become advantageous. Over time, they become so different genetically that they can't interbreed anymore, and they're considered separate species.
So they've evolved in different directions.
Exactly. And we should mention Alfred Russel Wallace, who came up with similar ideas about natural selection and speciation around the same time as Darwin.
Oh, right.
They even published some papers together, but Darwin's book had a much bigger impact.
Okay. So Darwin didn't know about genes or DNA. How did we figure that part out?
That's right. Darwin's theory didn't explain the mechanics of inheritance.
Okay.
It was Gregor Mendel, a monk, who did lots of experiments with pea plants and figured out how traits are passed down.
Right.
He worked out that offspring inherit hereditary units, which we now call genes, from each parent,
Okay.
and that these units come in different versions, alleles, and some are dominant and some are recessive.
Right.
But his work was largely ignored at the time,
Really? Why?
partly because it wasn't published in a well-known place, and partly because people didn't understand enough about cells and chromosomes yet.
So it was ahead of its time.
Exactly. It wasn't until the early 1900s that scientists rediscovered his work and realized that chromosomes behave like his hereditary units.
Okay.
And then they realized that genes are located on chromosomes. And then in 1953, they discovered the double helix structure of DNA, which finally explained how genetic information is stored, copied, and passed on.
That must have been a big moment.
It was. It brought together Mendel's laws of inheritance with the physical reality of DNA.
Amazing. And now we have tons of evidence to support the theory of evolution, right?
We do. The fossil record is one big piece of evidence. It shows us the remains of ancient organisms and how they've changed over time.
Right.
We can see how different groups of organisms are related and create evolutionary trees. And another strong piece of evidence is antibiotic resistance in bacteria.
Okay.
This is happening right now. Bacteria can develop resistance to antibiotics through random mutations.
Right.
When we use antibiotics, the non-resistant bacteria die, but the resistant ones survive and reproduce, passing on their resistance genes.
And that's how we get superbugs.
Exactly, like MRSA.
Right.
Bacteria reproduce really quickly, and mutations happen often. And because we use antibiotics so much, we're creating a situation where resistant bacteria have an advantage.
Right.
And then these resistant bacteria spread, and because we don't have immunity to them, and we might not have effective treatments, they're a big threat.
So what can we do about it?
Well, we need to be more careful about how we use antibiotics. Only use them when we really need them and make sure we finish the whole course.
Okay.
We also need to develop new antibiotics, which takes time and money.
Right.
And we need to be really careful about hygiene to stop the spread of resistant bacteria.
So it's a serious issue.
It is. Okay, lastly, let's talk about how scientists classify all the different organisms on Earth.
Okay, how do they do that?
So classification is how biologists group organisms based on their characteristics and evolutionary relationships.
Okay.
The Linnaean system, developed in the 1700s, groups organisms into a hierarchy of categories.
Right.
From broadest to most specific it goes. Kingdom, phylum, class, order, family, genus, and species.
Okay.
There's also the binomial system, which gives each species a two-part scientific name using the genus and species.
Right, like Homo sapiens for us.
Exactly. It's a universal way of naming organisms so everyone knows what we're talking about. More recently, we've got the three-domain system, which groups all life into archaea, bacteria, and eukaryota.
Okay.
This is based on new evidence mainly from studying ribosomal RNA. Archaea often live in extreme environments. Bacteria are the true bacteria. And eukaryota include everything with a nucleus in its cells, like protists, fungi, plants, and animals. And then we have evolutionary trees, which use all this classification data, along with anatomical and molecular data and fossil evidence to show how different organisms are related.
So it's like a big family tree for all life.
Exactly.
Well, we've covered a lot today, so